Hello everyone, welcome to the class. Today's topic of discussion is glycolysis. Glycolysis is a combination of two words, that is glyco plus lysis. Glyco means sugar, lysis means breakdown. So we can define glycolysis as a breakdown of sugar. Whatever we eat, that sugar molecules will be broken down into simpler products inside our body, right? And that breaking down of complex molecule into simpler molecule, we call it catabolism, right? And this catabolism and anabolism are metabolic pathway. There are two metabolic pathway, one is anabolism, other is catabolism. Anabolism means what? Whenever there is a synthesis or formation of complex molecule takes place from the simpler molecule, then we call it anabolism. Anabolism means basically the formation of simpler molecules from complex molecules, right? And catabolism is what? It is the breakdown of complex molecules into simpler molecule, right? So we can say that glycolysis is basically the breakdown of sugar. So we can give the example of glycolysis in the catabolism pathway, right? So glycolysis will be an example of catabolism, because here the breaking down of sugar molecules take place. So if we see glycolysis, what is the pathway? The glucose, glycolysis, glyco means sugar. So here glucose is the sugar, and the number of carbon in glucose is 6 carbon, right? So in this glycolysis process, the carbon is broken down into pyruvate. The pyruvate is having 3 carbon. The pyruvate is having 3 carbon and glucose is having 6 carbon. So how many molecules of pyruvate will form? 2 molecules. Then only it will be balanced, right? So here you can see 2 molecules of pyruvate will be formed. These 2 molecules of pyruvate will be formed because this is 6 carbon glucose and this is 3 carbon pyruvate, right? So for completion, for balancing, 2 molecules of pyruvate gets formed after the breakdown of sugar. So the entire glycolysis pathway is a 10-step pathway. Entire glycolysis pathway is what? It is a 10-step pathway. First 5 pathway we call the preparatory phase. First 5 pathway what we call? We call the preparatory phase because here ATP is used. ATP is used in the synthesis, right? And next 6 to 10 pathway. Next 6 to 10 pathway we call the payoff phase. Why we call it payoff phase? Because here ATP is released. So there are basically two pathways, for preparatory phase and payoff phase. In preparatory phase what happens? ATP is used up, and in payoff phase ATP is released. So if we the location of the glycolysis is cytosol, right? And glycolysis is basically a aerobic process, can takes place in anaerobic also, right? So this is basically what is the location of the glycolysis. So this is a process of glycolysis. Glucose is getting converted to glucose 6-phosphate. You can see. And here there is an addition of phosphate. And this phosphate molecule comes from ATP. Right? So you can see glucose is getting changed to glucose 6-phosphate. And this phosphate comes from ATP. Right? And glucose 6-phosphate again changed to fructose 6-phosphate. This you can see it is a step 1, step 2, step 3, 4, 5, 6, 7, 8, 9, 10. These are the steps, right? So what is happening? Glucose is getting converted to 6-phosphate by the addition of phosphate, and this phosphate comes from your ATP, right? And if you see here, glucose 6-phosphate is getting converted to fructose 6-phosphate. And fructose 6-phosphate again there is an addition of phosphate here. And this phosphate again comes from ATP and ADP. So you can see this pathway. If you observe this, I told you, right? First 5 pathway is a energy utilization. That means if you see this pathway first 5, here you can notice that there is a use of ATP. ATP is used up, right? And here after taking phosphate from ATP, the fructose 6-phosphate gets converted to fructose 1,6-bisphosphate. It is then changed to glyceraldehyde phosphate. This GLAP is nothing but it is your glyceraldehyde phosphate, right? This glyceraldehyde phosphate changes to 1,3-biphosphoglycerate. 1,3-biphosphoglycerate changes to 3-phosphoglycerate. This then changes to 2-phosphoglycerate, phosphoenol pyruvate, and this changes to pyruvate, right? So here if you say this is sixth process, and glyceraldehyde phosphate is getting converted to 1,3-biphosphoglycerate, right? Here what will happen, this 1,3-biphosphoglycerate now is releasing ATP. It is what it will do, it will form ATP. ATP is released. So as ATP is released, the bisphosphate gets converted to phosphate. Why? Because ATP is released. Phosphate molecule is released in the form of ATP. So here it gets changed to 3-phosphoglyceride. See, this 1,3 is the position where phosphate is attached. So you can say the first position where the first phosphate was attached is removed now, right? So it gets converted to 3-phosphoglycerate. 3-phosphoglycerate again gets converted to 2-phosphoglycerate. Now 2-phosphoglycerate, if you see again in the presence of enzyme enolase, change to phosphoenol pyruvate. Now phosphoenol pyruvate again releases ATP. Let me give another color for ATP release. Is again releases ATP. This is released, this is released. Again releases ATP. So here we can say that 2 molecules of ATP is released, that is 3 carbon product, that is your pyruvate is formed. 3 carbon product, pyruvate, right? So how many molecules of pyruvate will be formed? 2 molecules, because this is 3 carbon. 3 into 2 is your 6, so 2 molecules of pyruvate will be formed, right? So what we have seen in glycolysis, glycolysis is the breakdown of sugar, that is glucose. So glucose gets converted to glucose 6-phosphate. So here you can see addition of phosphate at 6th position, right? So this phosphate comes from ATP. So here ATP is used up. Again, glucose 6-phosphate gets structural changes. That's why we call it enzyme. We call it isomerase. It gets converted to fructose 6-phosphate. Now, fructose 6-phosphate, what it will do? It will again take phosphate from ATP and gets converted to fructose 1,6-bisphosphate. Right? Fructose 1,6-bisphosphate gets converted to glyceraldehyde phosphate, which then changes to 1,3-biphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and then pyruvate. If you see 1,3-bisphosphoglycerate while getting converted to 3, that means one position phosphate is being removed, right? So this phosphate is removed in the form of ATP. In the form of what? In the form of ATP. Right? So again here phosphoenol pyruvate will get converted to pyruvate. So this phosphate is removed. So here this is the second time where ATP is first ATP released here, and this is second ATP, which is second step where ATP is released is this position. Right? Yes, I think now it is clear to everyone. Glycolysis. So if you see what will happen, glucose will get converted. We have seen that glucose, this is what we have seen, glucose breaks down. So glucose is getting converted to your pyruvate. How many moles of pyruvate? 2 moles of pyruvate. Right? Now what this pyruvate will do once it is broken down? If the pyruvate, if the condition is aerobic, aerobic means what? Oxygen is present. If oxygen is present inside the cell, then it will move to acetyl coenzyme A. It gets converted to acetyl coenzyme A, which then enter into another cycle, which we call TCA cycle or Krebs cycle. This acetyl coenzyme A then enter into another cycle, which we call TCA cycle or Krebs cycle, and here it releases carbon dioxide and water as an end product. Right? This is what is the fate of pyruvate in case of aerobic condition whenever oxygen is present. If in case oxygen is absent, that is anaerobic condition. In case of anaerobic condition, it leads to the production of 2 molecules of lactate. It leads to the production of 2 molecules of lactate. That is what we call 3 carbon. Right? 2 molecules of lactate, it leads to the formation of 2 molecules of lactate in case of anaerobic condition. Again, in case of anaerobic condition in yeast, it leads to formation of ethanol and carbon dioxide. So what we have seen is that in the presence of oxygen, when oxygen is present, the glucose is getting converted to your pyruvate, and pyruvate is getting converted to acetyl coenzyme A. And this acetyl coenzyme A enters into the Krebs cycle and changes to carbon dioxide and water. But if oxygen is absent, in case if oxygen is absent, what will happen to this pyruvate? This pyruvate, in case if oxygen is absent, this pyruvate will changed into lactate. How many molecules of lactate? 2 molecules of lactate, right? And it will again change to your ethanol and carbon dioxide. So what you can see is, in absence of oxygen, pyruvate getting converted to lactate, ethanol and carbon dioxide. And in presence of oxygen, it gets converted to acetyl coenzyme A, which enters to your Krebs cycle, and end product of Krebs cycle is carbon dioxide and water. Right? This is the fate of pyruvate. So again, in detail, I have explained this stepwise so that you can understand it properly. If you see step 1, glucose utilizes ATP and ADP and gets converted to glucose 6-phosphate. So here there is a utilization of 1 ATP, right? If you see second step, glucose 6-phosphate is getting converted to fructose 6-phosphate with the help of enzyme phosphoglucoisomerase, right? So in the third step, fructose 6 is getting converted to 1,6-biphosphate. That means again addition of ATP takes place, right? And this ATP, again addition of ATP means what? Phosphate is added to the fructose 6-phosphate from ATP. Right? Now this fructose 1,6-bisphosphate is getting converted to glyceraldehyde 3-phosphate, and this is dihydroxyacetone phosphate. Right? So this is what we call glyceraldehyde 3-phosphate. And this is what we call dihydroacetone phosphate. Dihydroacetone phosphate, right? So this dihydroacetone phosphate changes to glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate again this again will go back to this step, you can say, right? So this glyceraldehyde 3-phosphate is getting converted to 1,3-biphosphoglycerate. Now here how many phosphate? 2 phosphate. Bi means 2 and 1,3 is the position where phosphate is attached. Now what will happen? There will be removal of 1 phosphate, and that 1 phosphate is removed in the form of ATP. So there is a production of 2 ATP, right? And 1,3-biphosphoglycerate changes to 3-phosphoglycerate. 3-phosphoglycerate then changes to 2-phosphoglycerate. 2-phosphoglycerate changes to phosphoenol pyruvate. And phosphoenol pyruvate again releases 2 ATP, gets converted to your pyruvate. So this is the reaction of glycolysis, right? So we will see the energy sheet now. How much energy? अगर मैं यहाँ पे फिर से ग्लाइकोलाइसिस के पाथवे को डिफाइन करूं, ग्लाइकोलाइसिस के स्टेप्स को, ताकि आपको क्लियर समझ में आए. पहले हमने क्या देखा था? जो ग्लूकोज था वो किस में चेंज हुआ था? वो चेंज हुआ था आपका ग्लूकोज 6-फॉस्फेट में, राइट? और ग्लूकोज 6-फॉस्फेट आपका किस में चेंज हुआ था? वो हुआ था फ्रुक्टोज 6-फॉस्फेट में. वो हुआ था फ्रुक्टोज 6-फॉस्फेट में, राइट? फ्रुक्टोज 6-फॉस्फेट किस में चेंज हुआ था? फ्रुक्टोज 1,6-बाइफॉस्फेट, राइट? फ्रुक्टोज 1,6-बाइफॉस्फेट चेंज हुआ था आपका ग्लिसरल्डिहाइड 3-फॉस्फेट, ग्लिसरल्डिहाइड 3-फॉस्फेट प्लस डाइहाइड्रोक्सी एसीटोन फॉस्फेट प्लस डाइहाइड्रोक्सी एसीटोन फॉस्फेट, राइट? अब यहाँ पे मैं पहले एनर्जी का आपको क्लियर, लेट मी फर्स्ट क्लियर द एनर्जी पार्ट हियर. So if you see the enzyme which is needed for glucose to change to glucose 6-phosphate enzyme is your hexokinase, right? Glucose 6-phosphate getting converted to fructose 6-phosphate with the help of enzyme phosphohexose isomerase. Phosphohexose phosphohexose isomerase, right? Fructose 6-phosphate changes to fructose 1,6-biphosphate with the help of aldolase. Fructose 1,6-biphosphate is then changes fructose 6-phosphate, it changes to fructose 1,6-phosphate. Sorry, I have written a wrong enzyme here. It should be your phosphofructokinase 1. Phosphofructokinase 1, right? Kinase means what? Whenever you get the word kinase, that means there is an addition of phosphate. See here, there is also hexokinase. Kinase means addition of phosphate. See, phosphate getting added to 6th position. Again here kinase. So here for addition of phosphate, right? Now fructose 1,6-biphosphate getting changed to glyceraldehyde 3-phosphate with the help of aldolase. With the help of what? With the help of aldolase, right? And this aldolase will convert fructose 1,6-biphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this first step, this is what? This is your first step, second step, third step, fourth step, fifth step. And now we will see the energy utilization. So in the first step, there is an addition of ATP. That means there is a addition of phosphate. That means ATP is utilized and it generates ADP, right? In the third step, again you can see fructose is getting converted to 1,6-biphosphate. That means one more addition of phosphate. So here you can see again ATP is utilized and it gets changed to ADP. Right everyone? Now, so here you can see as ATP is utilized. So this slide, the first 5 steps, what we call this? We call this preparatory phase. We call this what? We call this preparatory phase, right? Now we will deal the next part. Next part, if you say glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate. So how many molecules of glyceraldehyde 3-phosphate? Yes, there will be 2 molecules. Why 2 molecules? Because the dihydroxyacetone phosphate will also form glyceraldehyde, right? So here there will be 2 molecules. Now glyceraldehyde 3-phosphate gets converted to 1,3-biphosphoglycerate. 1,3-biphosphoglycerate, right? 1,3-biphosphoglycerate changed to your what? 3-phosphoglycerate. 3-phosphoglycerate, right? 3-phosphoglycerate changes to your 2-phosphoglycerate. 2-phosphoglycerate. 2-phosphoglycerate changes to phosphoenol pyruvate. Changes to phosphoenol pyruvate. And this phosphoenol pyruvate changes to your pyruvate, right? So this is the pathway. So here you can see there is 2 molecules of glyceraldehyde, 2 molecules of 1,3-biophosphor. From here 2 molecules started everywhere. 2 molecules and again pyruvate 2 molecules. 2 molecules. Now we will see the enzymes. So here glyceraldehyde 3-phosphate changes to 1,3-biphosphoglycerate. So there is a utilization of enzyme glyceraldehyde 3-phosphate dehydrogenase. What is the enzyme? It is dehydrogenase. genase, right? 1,3-biphosphoglycerate changes to phosphoglycerate, and that is what we call phosphoglycerase kinase, right? This is what we call what? Phosphoglycerase kinase. Phosphoglycerase kinase. Now 3-phosphoglycerate will change to your 2-phosphoglycerate with the help of enzyme mutase. With the help of enzyme mutase. And 2-phosphoglycerate gets converted to your phosphoenol pyruvate with the help of your enzyme enolase. Right? Phosphoenol pyruvate then changes to pyruvate with the help of enzyme pyruvate kinase. Right? Now there is a elimination of ATP, or you can say release of ATP. So this release of ATP we will see here, where the ATP is released, glyceraldehyde 3-phosphate getting converted to 1,3-biphosphoglycerate. So here in this step what will happen? 2-NAD positive will change to NADH. 2-NAD positive will change to 2-NADH. So here there is a, you can say, there is a release of 2-NADH molecule. That means this is also a form of energy. We will see that. Now 2-NADH, right? Clear everyone till here? Next, what will happen? When 1,3-biphosphoglycerate gets converted to 3-phosphoglycerate, again you can see from 1,3 it is changed to 3. So here also there is an elimination of ATP molecule, right? So here what will happen? 2 molecules of ADP gets converted to 2 ATP. Clear everyone? So here you can see there is again production of, this is first where 2-NADH produced, second 2 ATP is produced. Third step is your when phosphoenol pyruvate gets converted to pyruvate. In this step also you can say there is a elimination of ATP. 2 ADP getting converted to 2 ATP. So this is also a elimination of ATP. So this is the third part where ATP is being released. So if you say this is the first part, let me tell you how many steps the ATP and NADH are released. So if you see this is first part where when glyceraldehyde 3-phosphate changes to 1,3-biphosphoglycerate, there is a release of 2-NADH. Second, when 2 ATP is released, when 1,3-biphosphoglycerate changes to 3-phosphoglycerate. And this phosphoenol pyruvate, when released to pyruvate, there is a production of third ATP. Right? So that is what, this is what actually is the glycolysis process. Right? Now we will see the energy part, energy calculation part. Energy yield in glycolysis. Energy yield in glycolysis. If you say energy yield, now in the first step, if you remember, in the first step there is a utilization of first step. What will happen? ATP, you can write like this. In the first step, ATP is, in the first step, ATP is used, right? Again in the third step, ATP is used. So number of ATP used is equals to, therefore, number of ATP used is equals to 0, 2. You can see 2 molecules of ATP is being used, right? Now we will see ATP released. So how ATP is released? If we see this part, ATP released part. Now if you noticed, in which step? In the fifth step there is a release of. In the fifth step there is a release of NADH. How many molecules of NADH? 2 molecules of NADH, right? In the sixth step, there is a release of ATP. How many ATP? 2 molecules of ATP, right? And in the 10th step, 5th, 6th and 10th step, there is the release of what? Again 2 molecules of ATP, right? So, how many molecules of ATP are released? If we calculate in the form of ATP, this too you can see there is how many ATP? 4 ATP. Directly if you combine this two, there is 4 ATP, right? And from NADH, let me tell you here in this part, 1 NADH releases 1 NADH releases how many ATP? It releases 2.5 ATP, right? Now here how many? 2 NADH. So 2 NADH will release how much ATP? 2 NADH will release your 2 into 2.5 ATP. That is equals to 5 ATP, right? So here you can see from 2 NADH will be getting a 5 ATP, right? So total how much ATP you are getting? 9 ATP. So ATP released is 9 ATP. But out of 9 ATP, the number 2 ATPs are already being used. Yes, if you see out of 9 ATP, 2 ATP are already used. So if you minus 2 from 9, how much will be remaining ATP? 9 ATP is released and 2 ATP is used. So therefore, net gain of ATP or total ATP gain is equals to how much? 9 minus 2 ATP. 9 minus 2 is how much? 7 ATP. So from glycolysis, how much energy we are getting? We are getting 7 ATP. That is your energy from glyco, or you can write energy yield from glycolysis. That is energy yield from glycolysis, right? This is what is the energy we get from glycolysis. So whenever you eat food, you get energy. This is how your body gets energy. So if I give a quick revision of energy calculation part, quick review of energy calculation part, we have seen that in the preparatory phase, there is a utilization of 2 ATP. 2 ATP used up in the preparatory phase. And in the payoff phase, you can see how many ATP is released. In the payoff phase, we have seen in the payoff phase, we have seen there is a release of 2 NADH, 2 ATP and again 2 ATP. So total how much ATP is released? If we calculate here, total how much ATP is used? Total 9 ATP is released. Total 9 ATP is released. How 9? Because if I calculate here in this part, 1 NADH release 2.5 ATP, right? So 2 NADH will release 2 into 2.5, that is equals to 5 ATP, right? That is equals to how many ATP? 5 ATP. So from 2 NADH, how much you are getting? 5. So 5 plus 4, 9 ATP. Now we will see the net gain of ATP. Net energy yield. Net energy yield from glycolysis is equals to 9 minus 2 ATP. 9 minus 2 ATP is how much? it is equals to your 7 ATP. So this 7 ATP is the energy yield from glycolysis, right? So this is what is the quick review of the energy, and this is, you can say, it is very to the power 1000 important, right? So I think it is clear to you. If you have any doubts, please let me know. And thank you students for joining the session. Next class, we will be dealing with your Krebs cycle. Thank you so much students for watching it. Thank you.